And also, I've worked in the past as an epidemiologist for the state health department, where I got involved in food safety and specifically produce food safety. And then I went back and had got a PhD in microbiology and moved into research at the Western Center for Food Safety, which is sort of like your ARC. It's an academic center of excellence that works with industry and with our Food and Drug Administration to get real world practical solutions to food safety challenges. And because of what's been going on out here in the United States, and specifically California, where we're located, our focus has been produce food safety, especially at the pre-harvest level. And the background is that a lot of these pathogens that we're having problems with are actually from animal reservoirs rather than humans. Not to dismiss norovirus and hepatitis and other important human fecal-borne pathogens, but in the case of some of these emerging diseases, they are actually primarily bacteria from animals. And these can be wildlife or domestic animals, and it's not, the produce food safety is emerging, but it's not at least bit unknown that some of these pathogens that come from feces can get into the food supply directly through ingesting undercooked meat from animals, whether it be cattle, undercooked burgers, or undercooked deer meat, direct contact, such as in petting zoo settings. And then the topic today is this indirect fecal contamination through direct exposure or through indirect, such as water or bioaerosols. And these pathogens are tricky because they don't have a single reservoir. It's not like just dealing with hygiene in humans. There can be many different animals and groups of animals, including kangaroos, that can carry some of these. And there is a long list of examples of what we call this mouthful zoonotic enteric pathogens. And these are basically pathogens that often do not make the animal sick. So those cattle behind me are positive for E. coli 0157, and they are very healthy. They're on grass. They're not in factory farms or being in high density situations, but we can find 0157 in that setting as well as higher density settings for that particular pathogen, along with a number of these others. And this really brings up the concept that I know your center works on, and that's One Health, where that interface of animals, humans, and the environment, and how we as multidisciplinary professionals can work with industry, all the consumers all across the food supply to address issues. And produce is especially unique. I'm sure you've been talking about this today, but you know, the fact that the popularity out here in America, and I think across a lot of the developed world, is a strong interest and desire for raw products, including raw produce, which means there's no kill step. This isn't the spinach in the can from Popeye. This is raw product coming straight off of fields that are exposed potentially to animals and pathogens. This should be very rare, which it is, but the infectious dose is low. So it can only take a few cells to make a vulnerable person, like a young child, very sick. And there is some potential for pathogen attachment and risk for amplification across the food system. And we'll talk a little bit about that with an outbreak example I'm going to discuss. I wanted to bring up and highlight, hope you've all seen this. This was a collaboration with Robin and her group, putting together a fact sheet on the impact of animals and foodborne illness. And this is a figure that's included that's really helpful, just looking at some of these different sources. It's incredibly complex how these pathogens can get into the food supply. And so, you know, looking at outbreaks and preventive controls, you have to consider many different ways that these pathogens can get over to the actually into our plates, into our sets and such. So the way I'm going to illustrate this today, and I went back and forth, I have a lot of research data, not a lot of time. I've also been boots on the ground with outbreak investigations, and one of them was a watershed event. It really changed the face of produce food safety here in the United States. And since I was involved in this investigation, and it really defined how we are now doing our research for the last 15 years, plus how we've set our regulations, I'm going to run through some of the highlights from it. So I'll call this a case study of E. coli 0157, an outbreak that did a while ago, 2006. And honestly, when I was working for the state on this, I never would have imagined that this many years later, I'd still be talking about it, and it's still extremely relevant to what's going on, not only in our state, our country, and even worldwide with our concerns with our raw fresh produce. This is a picture from that outbreak when I first got down there. We went over to a deli and saw the sign. All spinach had been pulled off the shelves, everything. No, there was nothing in the grocery stores, the restaurants. It was a complete advisory to not eat spinach, which was just devastating for our agriculture. These fresh cut bag baby spinach were just huge in popularity. Juicing and health food was very big in the mid 2000s, and the industry could barely keep up. And we've long had a coexistence between animals and production ag, as well as produce production. And this particular region where we grow a lot of our leafy greens. You heard from Chana Rock this morning. The second largest area down in the Southwest desert to grow leafy greens, but the largest area is actually on the Central Coast, which is below San Francisco, if you've ever been out to California. And we have many specialty crops, from strawberries to leafy greens to artichokes. And we also have animal agriculture, primarily range cows like the ones behind me, not feedlots in this particular area, and a lot of wildlife corridors. So in this outbreak, we went down. It was not new. There'd been some outbreaks, probably about over a dozen smaller outbreaks traced back to lettuce and a couple to spinach. And so we were pretty quick to be sent down in teams with FDA and the state and sample. So we're like, what are we going to sample? This is the actual ground zero. We're at the ranch where we eventually found the outbreak strain, but not in the actual produce field. It had already been plowed and tilled, and it'd been, you know, a couple months before you actually identify and get down on these farms. And so a lot of the spinach was already gone. We were just looking at bare dirt. So you start looking around at irrigation water and other sources on the farm. All the irrigation in this area, virtually all of it is deep well. It's not like down in the Southwest desert with the big canals where you would treat water. This is underground water that rarely tests positive for pathogens or even generic E. coli. And so that, you know, becomes a concern. How could this underground water have gotten contaminated if it was the source? And so what we start to do is pull up a Google map and look at the adjacent land use. What else is going on this farm? And the spinach field is kind of in the center of this picture. And it's literally that earlier, just a classic look from this area where you have, when you get into some of the foothill areas, you'll have riparian habitats, rivers, wildlife corridors, a lot of vineyards around these produce growing areas, as well as cattle ranches. And these cattle ranches, again, this was the one that was adjacent about a mile away from this implicated spinach field, was grass-fed beef. It was not grain-fed. These were grass-fed, marketed grass-fed cattle on irrigated pasture. And they did have access to surface water. The surface water was not being used for irrigation, but it did have some potential to leach into older ag wells. So that was a concern. The other concern was, again, this as we looked around this property, we saw a lot of feral pigs, and I believe you have issues with feral pigs in Australia, and it's a big problem out here in the United States. They were actually imported, and the Eurasian wild boars were brought into California for game and interbred with our domestic pigs, and they are very mobile, very common in produce growing area. And in this farm, we did see evidence of animal intrusion into this abandoned spinach field and defecation. Ultimately, we found the outbreak strain in multiple locations all over this ranch, and in the cattle feces nearby, but a mile away. This is the dry season. There was no runoff, no rain, no direct contact between the cattle and the fields. We did have wildlife moving through. We found this strain in the feral pigs, surface water, river sediment, pasture soil, and it matched the human stool and bag spinach. So it was the same as the clinical specimens that were collected. And so what we ended up learning was that E. coli could be an issue. We knew it was an issue in cattle as a primary reservoir, but I did not have a good idea back in 2006 whether it was spilling over to other wildlife. There was some evidence that it spilled over to deer, which are another ruminant, but this showed that feral pigs were another potential reservoir. And there were a lot of potential sources. And some of the hypotheses, there was never an absolute smoking cow, so to speak, but some concerns were that there could have been cross-contamination and growth of the pathogen. It might have come in from animal feces in a small amount and then grew as it moved through the production chain, but also the concern that other practices could have amplified it, such as using soil amendments, compost teas, equipment. And so in the end, we did see that there were a lot of potential risk factors, and this just really set off a huge effort of voluntary and eventually regulatory good agriculture practices for the industry. And there's been just a ton of science and people involved in this effort out here in the United States. I will point out that although this did have a cattle wildlife link, there was later in 2011 a smaller outbreak associated with fresh strawberries at a local farmers market and Upick farm up in Oregon, which is more in the North State, and this outbreak strain was found in wild deer that were getting into the strawberry fields. You can see their fecal material, and there were no cattle nearby. There was never any indication of a cattle link, and so it's just different every time, and so you can't make assumptions on these, and that's where we get into the research and what we call ecological epidemiological studies, and a lot of these are longitudinal, multi-year, and we're just starting a new one right now with FDA that's going to go 3 years over in this area where the cows are behind me, to again get down there and look where we know that we have endemic E. coli 0157 and other Shiga toxin E. coli in this area, as well as Salmonella, but we don't totally understand is how it's moving from potentially distant locations over into these fields. And if it's coming in at a low level, how is it amplifying and causing a national or international outbreak? And so we've done a lot of work. I don't have time to run through all of our different research, but I think a lot of you know Trevor Suslow with PMA, and this was a paper we published earlier this year looking at wild birds that were actually commingling with cattle, and a lot of concerns about birds because they can be very a big nuisance. I think talking to Robin, you have that in Australia as well, and our species are probably different than what you deal with, but what we find, not surprisingly, is that the solitary native birds are rarely, if ever, do we isolate any pathogens, but when you get into these birds that flock and aggregate and are nuisance, like the starlings, blackbirds, sparrows, Canada geese, we do find the pathogens, not necessarily at high levels, but if you've got 10,000 starlings in a field or 200 Canada geese pooping, that risk goes way up. And so we're really looking at, how do we co-manage this problem? How do we still protect the environment? How do we protect the crops? A lot of our approach is to, actually walk the fields and look for animal intrusion, cordon off the area. Don't send that to market. That's the most obvious preventive measure, and usually will work. And it's if there's gross contamination, and that's now in our regulations, and if that's not harvested, but it gets harder when you have more subtle contamination events, or if there's something happening in the environment to amplify the bacteria. And we've tried different methods. This picture in my previous slide is a falconer. We actually brought a falconer out to help chase off nuisance birds that were flocking and landing in lettuce and broccoli fields, and we showed some efficacy. we're trying to use a combination of methods that that fit what we call co-management, which is an approach to minimize these contamination hazard that I consider one of these low probability, high consequence events, especially when it comes to the potential for wildlife intrusion. It's not likely to happen, but if it does, I mean, you can have children on dialysis machines, and it's just such a horrible pathogen with E. coli 0157, and then what it does to the industry as well. That we have to address it. It can't be ignored, but we also have to balance that with conservation efforts in our ag lands, and more recently, this idea of good ag neighbors. Because of some of the recent outbreaks, there have been in the media and the public perceptions toward cattlemen have become really negative, and the cattlemen are feeling, very concerned about their livelihood and their ability to continue to produce next to produce fields, and we didn't previously have problems. So something new has happened in this environment that is still being unraveled, because again, these aren't giant farms with runoff and rain. We're growing produce in the dry season, and as Chana might have talked about, with the water treatment, maybe that's what we need, some kind of uh, ability to address these pathogens in the environment without shutting down our other animal ag and our conservation efforts with wildlife. So with that, I'm going to finish and see if there are any questions. And I have so many people to thank. I just ended up throwing, this is my lab, some of my lab staff, and we're doing some field trials with E. coli surrogates, and this one is actually with raw manures and looking at persistence of pathogens. And so I tweet, and I put this on Twitter, and NPR picked it up and did a little story on my lab, and they had a lot of fun with it. So thanks for listening, and I'll stop here and see if we have a minute or for any questions.
Thanks.